This project was independently produced by the team at ***. We want to create a user-supported model for podcasts that doesn't rely on advertisers. So if you enjoy this series, please consider a small donation to our Patreon account, which can be found at patreon.com/**media. Thank you. I have uncovered exotic and deadly bacteria that have hitched rides out of high-security labs on workers' dirty clothing, silently spreading contagion for weeks. I have revealed how spacesuit-like protective gear and tubes carrying safe oxygen to scientists have torn or broken repeatedly, and high-tech safety systems have failed dramatically. Vials of viruses and bacteria have gone missing. Researchers bitten by infected lab animals have been allowed to move about in public rather than being quarantined while waiting for signs of infection to appear. These and similar safety lapses are happening with disturbing regularity at elite U.S. labs operated by government agencies, the military, universities, and private firms. There is no reason to believe they aren't happening at labs in other countries as well. The notion that more than 2.7 million deaths worldwide so far could be the result of a lab accident has been met with skepticism and derision by many journalists and scientists, who often portray it as a crackpot conspiracy theory fueled by former President Donald Trump's China-bashing rhetoric. Without question, the lab leak theory has been politically and racially weaponized in ugly ways. Nonetheless, that rhetoric needs to be separated from legitimate questions about lab safety that are deserving of investigation. These are the words of veteran investigative journalist Allison Young, quoted from her USA Today opinion piece titled, "Could an accident have caused COVID-19? Why the Wuhan lab leak theory shouldn't be dismissed." Young is correct in noting that association with President Donald Trump kept many serious scientists and journalists from being willing to step anywhere close to considering that the SARS-CoV-2 virus escaped from the Wuhan Institute of Virology. But with Trump no longer president, more and more researchers, reporters, and politicians are willing to take seriously the almost unbelievable coincidence that a novel, bat-borne, SARS-type coronavirus would begin spreading amongst the urban population in the city that just happened to host the research of China's most prominent bat-borne SARS-type coronavirus expert, Dr. Shi Zhengli of the Wuhan Institute of Virology.
In China, the city of Wuhan is still on lockdown, with Americans trapped inside as the number of worldwide cases
in Italy has surged by 919 in a single day. That is more than
doubling. Scientist Tulio de Oliveira discovered the new variant after observing a dramatic
is acting as an accelerant to conspiracy theories. The digital era we live in
came as the United States recorded more than 2,400 deaths in just
that has been reported from Brazil. And the third has come as the more contagious variant of COVID-19
the number of deaths recorded from COVID in the UK has passed
market in Wuhan, China, show it likely played a role in the development of
China's study into the origins of COVID-19. They've concluded that it's unlikely it came from a Wuhan lab.
I'm John Duffy, and this is Origins, Birth of a Pandemic, Part 2.
The very basic thing about a furin cleavage site is that it is a protein sequence that allows proteases, which are basically enzymes that cut other proteins. This cleavage site allows these proteases to cut the spike protein of the coronavirus in a particular way that it enhances its infection, especially towards human lung.
This is Dr. Jonathan Latham again. We heard a lot from him in our first installment of this series. He is a virologist and co-founder of the Bioscience Resource Project, as well as Independent Science News. Right now, we're talking about something called a furin cleavage site, which is a particular piece of the SARS-CoV-2 virus.
It's also considered to be responsible for allowing the virus to enter nerve cells, to enter gastric tissues, to basically spread around the body in a way that most coronaviruses cannot. So it plays a very key role in the infection process. This furin cleavage site is super important to understand because when you think about the evolution of coronaviruses, what you naturally do, you think, what were the parental viruses? Who were the ancestors? One thing that we know is that in general, coronaviruses from a particular species will readily recombine with coronaviruses from the same species. They're less likely to recombine with more distantly related coronaviruses. For example, some alpha coronaviruses, they in general do not recombine with beta coronaviruses. And so you ask an interesting question about SARS-CoV-2. You say, where did it get the furin cleavage site from? And the answer is, we have no idea. The nearest viruses that have furin cleavage sites, firstly, don't really look like this furin cleavage site, and so there's no obvious ancestor. So the explanation for that then becomes narrowed quite significantly between some kind of unusual evolutionary event in which the furin cleavage site derived by some unlikely set of mutations, or you arrive at the possibility that it was put there by a person, maybe at the Wuhan Institute of Virology. Most of the speculation around where this furin cleavage site came from has to do with the fact that it appears to favor the idea of a lab origin.
Researchers have pointed to the fact that other coronaviruses have furin cleavage sites, and have tried to say that this means there is no reason to suspect that its presence in SARS-CoV-2 is indicative of a laboratory escape. But it should be noted as well that humans have added a furin cleavage site to the SARS-1 virus. Back in 2006, scientists at the University of Montana did just this, and published a paper titled Furin Cleavage of the SARS Coronavirus Spike Glycoprotein Enhances Cell-to-Cell Fusion, but does not affect virion entry.
SARS-CoV-2 is not a bat-infecting virus, and this is also an argument against it having a zoonotic origin. You would expect a virus that came from bats, it should still be able to infect bats. It's not clear whether the furin cleavage site is the reason why the virus does not do well in bats. It seems like the spike protein is not well adapted to the bat ACE2 receptor. So that's one more example why this may be an engineered type virus.
When people hear the phrase engineered virus, they are likely to start thinking that this implies some sort of shadowy project to intentionally create a plague virus to release upon the global population. But the reality is that there is a whole type of viral research called gain of function. The following audio is from a discussion hosted and then uploaded to the internet by the Center for Infectious Disease Dynamics back in December of 2016. The topic of the discussion is the possible risks and potential rewards of gain-of-function research.
This is research that adds functionality and in fact oftentimes increases virulence of existing pathogens to allow us to study them and study the characteristics they have when they're now worse off, right? So many of us can immediately, from a sort of sci-fi standpoint, imagine what the costs and the fears associated with these experiments are.
The rationale is pretty simple. If you can identify a specific part, or say a gene in an organism that is responsible for disease, then you actually have a target that you can aim for therapeutics. The transmission experiments get you into another realm because they're really changing the biology of the virus.
We as a scientific community don't know what we're doing, so it's really important to, it's true, I know. There was recently the formation of the Cambridge Working Group. Marcel Salaté is part of that, and they gathered together a whole range of scientists to figure out what might we do, because we really don't know whether labs should work on this and how and to what extent government authorities should step in and mandate that this work shouldn't be done, because it is absolutely clear that it's very dangerous work.
There is obviously a very strong argument for gain-of-function experiments. I've got no issue with that. The question is whether we should do it where we're trying to enhance certain types of function, in this case mammalian transmissibility, in pathogens that we're genuinely very scared about. We have every reason to be very scared. And I think this discussion is an example of why we need a moratorium here. We need a pause to think, what sort of questions are we really going to get good things out of for taking these risks? And that has to be put up against the possibility these things get out. And you can measure the rate at which people lose pathogens. I mean, the pathogens get out of labs, and it's not zero.
And this is clearly a discussion that's going to be continuing in the scientific community for some time to come.
The speaker who said that we in the scientific community don't know what we're doing, a line which elicited nervous laughter from some of the other participants, mentioned the Cambridge Working Group, which formed in 2014. According to their website, the Cambridge Working Group, quote, consists of concerned scientists and experts in legal, ethical, and other dimensions of potential pandemic pathogen research. And it formed, quote, to coordinate efforts to enhance public understanding of the biosafety risks of such research. Marc Lipsitch, a professor of epidemiology at Harvard's University School of Public Health, is a founding member of the Cambridge Working Group, and he has been a critic of gain-of-function research for many years.
The kinds of experiments that I'm describing are similar in their potential for mishap to, I would argue, the most dangerous kinds of experiments that we have going on in any laboratories, biological laboratories, in the sense that they are creating strains that have high virulence, that have transmissibility and pandemic potential, and are novel. So they fulfill those three requirements of a pandemic pathogen. And the only other type of work that I would say matches that is work on smallpox.
This audio comes from a presentation hosted by the Center for the Study of Existential Risks at Cambridge University. This talk happened back in early 2015, right after there had been a moratorium on funding gain-of-function research by the US government via the National Institutes of Health.
Fast forwarding to October, there was an announcement from the White House that the US government was going to pause funding on all such work for a year while a risk and benefit assessment was done. That funding pause generated letters to recipients of 18 grants from the National Institutes of Health. 18 out of approximately 250 grants that the US government was funding at the time on MERS, SARS, and flu. We didn't expect MERS and SARS to be in it. And in fact, those two coronaviruses were removed from the funding pause in December.
What Lipsitch noted at the end of that clip is that when this funding pause on gain-of-function research happened back during the Obama administration, in October of 2014, it turned out there were active grants that had been approved by the NIH to do gain-of-function research on both MERS and SARS-1. Lipsitch mentions that the work on MERS and SARS was actually allowed to continue. It turns out that technically, the moratorium on work that was already funded and underway was voluntary. And the moratorium was officially ended entirely in December of 2017.
There have been a number of major breaches that have caused large, not existential, but large real risks or potential risks. In Beijing, there was a laboratory accident with SARS about a decade ago, in which there were 2 primary cases who went on to infect 7 others before it was controlled. There were similar events in Taiwan and Singapore, also with SARS virus, but there were no secondary cases in those examples. There was the famous foot and mouth disease release from Purebright that you're aware of if you've been in the UK since 2007, which caused major agricultural consequences, and probably the 1977 H1N1 strain that circulated for 22 years following that reemerged because of a laboratory escape, although that is not proven.
For Professor Lipsitch, the concern isn't any and all work with deadly viruses. The concern is adding virulence to pathogens with pandemic potential. This would include work with SARS-type coronaviruses, such as the work done by Ralph Baric at the University of North Carolina. Here is Baric giving a talk at the University of North Carolina School of Public Health.
I study coronaviruses, noroviruses, flaviviruses, and do some influenza virus research, just to give you some background of what I do. Since I've been on the planet, there's been an epidemic virus every decade, starting with Asian flu in 1957, Hong Kong in 1967 or 68. H1N1 came back in the 70s, followed by HIV, followed by West Nile, and then we got to the 21st century. And things sped up. SARS was the first epidemic virus of the 21st century, about 8,000 cases in 33 countries with a 10% mortality. Chikungunya virus emerged around 2005, the Mexican flu in 2009, MERS coronavirus emerged in 2012. It's still around today with a 36% mortality and about 2,200 cases. About the same time, H7N9 emerged in Southeast Asia, followed by Ebola with about 30,000 cases and a 39% mortality. And then Zika virus sort of drifted across Asia, ended up in Polynesia, and then arrived in the New World to cause another huge epidemic. So if you look at virus families, clearly the most dangerous viruses on the planet are coronaviruses.
Dr. Baric is a colleague of Dr. Shi-Zheng Li at the Wuhan Institute of Virology. And in 2015, Dr. Baric worked with Dr. Shi to create a chimeric virus in which they took the spike protein of a bat coronavirus and added it to a SARS-1 backbone. This whole issue of NIH funding for gain-of-function research and Dr. Ralph Baric's cooperation with Dr. Shi-Zheng Li would get major public visibility long after the COVID-19 pandemic began, when in May of 2021, at a Senate committee hearing, Senator Rand Paul of Kentucky grilled NIAID Director and public face of pandemic mitigation in the U.S., Dr. Anthony Fauci.
Dr. Fauci, we don't know whether the pandemic started in a lab in Wuhan or evolved naturally, but we should want to know. Instead, government authorities, self-interested in continuing gain-of-function research, say there's nothing to see here. To arrive at the truth, the U.S. government should admit that the Wuhan Virology Institute was experimenting to enhance the coronavirus's ability to infect humans. For years, Dr. Ralph Baric, a virologist in the U.S., has been collaborating with Dr. Shi-Zheng Li of the Wuhan Virology Institute, sharing his discoveries about how to create superviruses. This gain-of-function research has been funded by the NIH. The collaboration between the U.S. and the Wuhan Virology Institute continues. Dr. Fauci, do you still support funding of the NIH funding of the lab in Wuhan?
Senator Paul, with all due respect, you are entirely and completely incorrect that the NIH has not ever and does not now fund gain-of-function research in the Wuhan Institute
Do they fund
of Virology.
Dr. Baric?
We do not fund.
Do you fund Dr. Baric's gain-of-function research?
Dr. Baric does not doing gain-of-function research, and if it is, it's according to the guidelines, and it is being conducted in North Carolina.
Government defenders of gain-of-function, such as yourself, say that COVID-19 mutations were random and not designed by man. But interestingly, the technique that Dr. Baric developed forces mutations by serial passage through cell culture that the mutations appear to be natural. In fact, Dr. Baric named the technique the no-see-em technique because the mutations appear naturally. Nicholas Baker in the New York Magazine said nobody would know if the virus had been fabricated in a laboratory or grown in nature. Dr. Baric, an advocate of gain-of-function research, admits the main problem that the Institute of Virology has is the outbreak occurred in close proximity. What are the odds? Baric responded, could you rule out a laboratory escape? The answer in this case is probably not.
I fully agree that you should investigate where the virus came from, but again, we have not funded gain-of-function research on this virus in the Wuhan Institute of Virology, no matter how many times
you're parsing
you say
words.
it.
There was
didn't
research.
happen.
There was research done with Dr. Shi and Dr. Baric. They have collaborated on gain-of-function research where they enhanced the SARS virus to infect human airway cells, and they did it by merging a new spike protein on it. That is gain-of-function. That was joint research between the Wuhan Institute and Dr. Baric. You can't deny it.
Dr. Fauci was, as Senator Paul suggested, parsing words. NIH funding doesn't go directly to the Wuhan Institute of Virology, but would move through intermediaries, such as the EcoHealth Alliance, or even indirectly when it funds the work Dr. Baric does in concert with Dr. Shi-Zheng Li.
Ralph Baric is a researcher at the University of North Carolina who specializes in coronaviruses. So Ralph Baric is firstly a collaborator of Zheng Li Shi, And secondly, he's been involved in some, in some ways very interesting and very scientifically competent gain-of-function research, but at the same time, a very alarming idea that you would take these viruses and basically humanize them and make them adapted to either humans or to mice, and then find that you've basically developed viruses that have extremely harmful properties.
There are different techniques used in gain-of-function experiments with viruses. One of them is serial passaging. This is basically when a virus is placed into a host, either a cell culture or a live animal. And before the immune system of that host kills the virus entirely, it's extracted and then placed into the next host. Doing this over and over and over again allows the virus to slowly adapt to that particular type of host. Aside from passaging, there are also more direct gene editing techniques, which Dr. Latham simply calls cutting and pasting. Since Dr. Latham believes that SARS-CoV-2 escaped from the Wuhan Institute of Virology, I asked him if he thought Dr. Shi Zhengli and her team had modified RATG13, the virus that Dr. Shi's team found in the mine in Mozhang that is the closest known relative of SARS-CoV-2.
I would be shocked to find out that the only explanation was cutting and pasting. The problem with proposing that SARS-CoV-2 arose from cutting and pasting would be that we don't have the building blocks to make SARS-CoV-2. Nature does its own cutting and pasting by recombination. If humans were cutting and pasting in the lab in the same kind of a way, unless all of those building blocks are hidden in the freezers of the Wuhan Institute of Virology, we have no idea where they come from. The simple idea of cut and paste would be that they took RATG13 as a backbone and they added a spike protein from a mystery virus that they have in their freezers. But what that would look like is that there would be a part of SARS-CoV-2 that looked basically identical to RATG13, and then other parts that were clearly different. Because the genome is so large, there would be signs that there were certain parts that had obvious provenance of viruses we know, and certain parts that did not. But also, for a person or a lab engineer to cut and paste a virus, and then to have that virus be perfectly adapted to humans when it came from mice, or it came from bats, or it came from experiments on primates or something like that, it would be really hard to build a virus that actually was perfectly adapted to people. If your virus escaped, it would still have to go through an evolutionary process. Like if a person was doing it, the only way that they could do that would be to basically passage the virus inside human cells for a very long period of time to make sure that every part of the virus was perfectly adapted to people. But even passaging in human cells does not adapt a virus to the human immune system. The human immune system has different requirements of a virus than does a cell culture. So if you perfectly adapted the virus in a human cell culture and then you put that virus into humans, well, it's not necessarily adapted to the human lung tissues versus the airway tissues versus the other tissues that it needs to enter. Whereas the virus that appeared in Wuhan in late 2019 appears to meet all those criteria.
So, according to Dr. Latham, cutting and pasting together SARS-CoV-2 from RATG13, or even a similar virus that perhaps Dr. Shi had in her lab, wouldn't really be possible because they would need all of the proper building blocks, and then that virus would not be as well adapted to the human immune system as SARS-CoV-2 has proven to be, right from the outset. And even serial passaging of RATG13 in human cells or in mice with humanized lung tissues, yes, that's a thing, would not likely result in the SARS-CoV-2 that we have witnessed arising out of Wuhan in December 2019.
The possibility that we have proposed is that what happened to cause RATG13 to become SARS-2 is that it evolved in the bodies of the miners. So the miners who were involved in the outbreak were ill for a long time. Some died more or less straight away, but some of them were basically in the hospital in an immunocompromised state. and were basically harboring the bat coronavirus that they had contracted in the mine. And they had that virus for around 5 months. We think that you can explain the jump between RATG13 and SARS-2 by the evolution of the virus inside the lungs of those miners. And what's interesting about this thesis is that it explains why SARS-2 is a virus that favors the human lung. So one of the things that needs explaining about SARS-2 is why does it infect the lungs, right? Why would a virus that came from bats infect human lungs when, for the most part in bats, these viruses affect the gastrointestinal system? That requires an explanation, right? Why does it bind so closely to the ACE2 receptor? Why does it spread so easily between humans? Why does it cause such a severe infection in humans? This is something that needs explaining. In the first few months of its existence in humans, even though it affected tens of thousands of people, barely evolved at all.
If Dr. Shi Zhengli and her team went into the mine in Mojiang to take samples from bats and bat guano, I asked Dr. Latham how a virus that evolved in the miners themselves would have made it into the possession of the Wuhan Institute of Virology.
Many samples from the miners are taken. The doctors took a thymus, they took blood samples, they took throat swabs. These samples were sent to labs all over China, but most specifically they were sent to the Wuhan Institute of Virology. And once they arrived at the Wuhan Institute of Virology, what would have happened to those samples is the normal process of isolating a coronavirus. You try to sequence the genome, you try to isolate the virus in some way, you try to culture the virus in some way. So essentially what we think happened is that in the subsequent research program, at some point, the virus that evolved in the miners basically escaped. And it may have been in the process of manipulation or making a vaccine or something, but it's also possible that no manipulation whatsoever was done with the virus that the Wuhan Institute of Virology got from the miners.
Dr. Latham explained what made the conditions in the miners' lungs different from those of an average person who comes into contact with a wild bat virus or even human-ready SARS-CoV-2.
A miner who shovels bat guano is probably going to be intaking a lot of dust. They may also have damaged lungs from previous mining work that they did or physical work. They're also going to be breathing deeply bat guano, which is full of like sharp insect body parts and so forth. And one of the super interesting things about the mine is that it appears to be a gold mine of coronaviruses. And what we know about coronaviruses is that they recombine super easily with each other. So if you put two different coronaviruses in one cell, they will automatically recombine with each other, and you will have basically a third coronavirus or a fourth or a fifth or a sixth coronavirus will evolve inside a single cell. What happened in the miners is they're taking in all this dust, all this guano, and it's penetrating deep into their lungs. And so they get a massive exposure. The miners get sick because they're inhaling a large quantity of basically a badly adapted virus. But as the virus infection inside them proceeds, there's a huge selection pressure for the virus to evolve inside those miners. You know, whenever you put an ill-adapted virus into a cell type or a tissue culture, what happens is the virus evolves to become more adapted to that environment. So it's continually mutating. And you have to imagine that there's these pockets of infection that are all over the miner's lungs. You know, a virus that evolves in one cell group or one little corner of the lung becomes better adapted than all the others, and it spreads around to other parts of the lung. But it can also hook up with other viruses that have evolved in their own particular way to become adapted to the lungs. And then what happens is recombination can kick in. A mutation, advantageous mutation that arises in one part of the lung in one virus lineage can basically recombine with another advantageous mutation that arises in another part of the lung. And this is very different from If you get a coronavirus at a party, what the person who gives it to you will have basically a monoculture of coronavirus. The virus that you get may evolve a tiny bit inside you, or it may have evolved a tiny bit inside them, but what you're getting is a monoculture. They're all identical to each other. So essentially, you have a very different infectious process going on, a very different process of adaptation, and a whole set of different circumstances from the ordinary situation of a coronavirus infection. What might normally take 30 or 40 or 50 years could actually happen in 6 months in the miner's lung.
If you recall, the miners in Mojiang who became ill with a mystery pneumonia all became sick starting in the spring of 2012. So I asked Dr. Latham why there would be a 7-year gap between those events and the beginning of the COVID-19 pandemic.
I think that's an important question to ask. One possibility is that they had difficulty isolating the virus from the miners' tissues. Another possibility is that they did have what amounted to an infectious human virus. But in order to study it properly, what they would have needed to do was have some very biosecure environment in which to do that. So you have to remember that at the time, China had no BSL-4 type labs. So those are the highest biosecurity labs. And the Wuhan Institute of Virology built such a lab in late 2017, early 2018.
This BSL-4 lab that was built at the Wuhan Institute of Virology was the subject of American State Department cables, first publicized by journalist Josh Rogan, that warned of, quote, a serious shortage of appropriately trained technicians and investigators needed to safely operate this high containment laboratory.
One possibility is they were waiting to do those kinds of human cell line experiments for the advent of their new BSL-4 lab. Perhaps even that's what one of the thoughts behind that BSL-4 lab was, to try to make use of this virus. One of the mysteries of all this is why they will publish a paper in 2016 not mentioning the miners' outbreak. There's something that they could assert that would point towards the usefulness of their research. Why would they not mention the miners' outbreak? There is something strange going on, even before the pandemic broke out. There are possibilities of what they were doing with that virus that are probably not fun to discuss. You know, there was some military involvement, or some group wanted them to keep quiet about this research. That's one possibility. But the other is simple technical difficulties or biological difficulties. Those are the seeming obvious explanations of why nothing would come out at that time.
So Dr. Latham and his partner, Dr. Wilson, at the Bioscience Resource Project propose a hypothesis that RATG13, as well as several other coronaviruses, made it deep into the damaged lungs of the 6 miners in Mojiang back in 2012, and that in the lungs of one of these miners, again, 3 of them died rather quickly, and the other 3 survived after being ill for 5 or so months, what we now know as SARS-CoV-2 evolved. It is known, thanks to the master's and PhD theses, that several sample types were taken from these men while they were sick in hospital, and that these samples were sent to various labs in China, including specifically the Wuhan Institute of Virology. And Dr. Latham suggests the possibility that either technical difficulty or even the need for a BSL-4 lab, which they wouldn't have for 5 more years, was why it took 7 years for the virus to leak from the lab. A second possibility remains, however. If the virus or viruses cultured from the samples taken from the miners were not fully evolved SARS-CoV-2, they could have been very close, closer than RATG13, and the final work needed to make them into SARS-CoV-2 could have been done by Dr. Shi Zhengli and her team, whether through gene editing or serial passaging. To know exactly what viruses and samples Dr. Shi and her team were working with, we would have to access the databases of the Wuhan Institute of Virology. But unfortunately, those databases were taken offline in September of 2019. This was brought to the attention of D.R.A.S.T.I.C by a man in the UK named Charles Small, who then joined up with D.R.A.S.T.I.C to explore the issue more deeply. This is Gilles Demaneuf again, the data scientist and D.R.A.S.T.I.C member we heard from in part 1 of this series.
He had found out basically that this database had gone off on the 12th of September 2019 because he was able to access the actual logs. It's not just one database, it's actually something like 15 databases. And we started basically looking at this into more details.
When asked about the databases being taken offline, Dr. Shi told the BBC the databases had been taken offline for security reasons after their website and emails had been hacked.
Well, no, she said that they took them off because of hacking attempts during the pandemic. Then a few months later, we found out that actually the date they took it off was the 12th of September 2019. So it didn't make any sense. Her answer doesn't make any sense at all, unless the pandemic started in September.
Dr. Shi then went on to say, quote, "All our research results are published in English journals in the form of papers. Virus sequences are saved in the GenBank database too. It's completely transparent. We have nothing to hide."
That's absolutely not true. That's not the way things work. First of all, in that database, there's not only viruses, sequences, there's also information about sampling. From the samples, they will do typically maybe a quick sequence of part of the virus, which basically is used as an index to quickly see what's there. And maybe they will do a full sequence, which typically is a consensus sequence. And that may end up in a database. But what is sure is that unless they publish something about the virus, unless they actually publish something about it, they do not put it in a public section. And there's a very good reason for that, is that they don't want someone to go and write something about the virus they put in a public section. If someone goes there and writes about it before them, they're at a loss. And there are many viruses that they didn't write about. And we keep finding more.
I asked Mr. Demaneuf if there was any indication that the Wuhan Institute of Virology was working with the samples from the Mojang miners based on what they could see in what remained of the Institute's logs.
We can't tell you for sure, because it's public, is that there's something like 30 samples from these miners at the WIV. Now, there's a bit of a confusion on the numbers. I think the Xi Zhengli basically disclosed 13 samples from these miners, but a more recent paper that was found by the seeker actually proves that the number is 30. And these samples are there, so they could be tested today, certainly, with more precise tests to try to find out what kind of bad code was likely. The WIV is sitting on important samples and is not disclosing them. That in itself is rather annoying. I don't see their interest in doing so. They should just say, here are the samples, do whatever test you want to do, and let's finish with that story, But they're not doing that.
According to the UK paper, the Daily Mail, quote, "In November 2019, at a time when US intelligence points to a potential COVID-19 outbreak at the lab, the Wuhan lab filed a patent for a device to treat injuries sustained while working with pathogenic viruses in a biosafety lab. The specialist tourniquet device, designed to wrap around the finger of someone who bleeds in a virology lab accident, appears to be the only one of several hundred publicly available patents which relates to the treatment of injuries." Certainly, this evidence is circumstantial, but let's put it all side by side. Starting in early 2018, the Wuhan Institute of Virology has a BSL-4 lab that comes online, and they are already in possession of the samples taken from the miners in Mojang. And then in September of 2019, we have the main database of samples and viral sequences mysteriously being taken offline. Then we have intelligence seen by NBC in the UK that shows in October of 2019 there is a period of days where there is no cell phone service in certain regions of the Wuhan Institute of Virology. The next month, in November, two interesting things happen. For one, researchers at the Institute are applying for patents for finger tourniquets that could be used by someone who is bitten on the finger by a test animal. Also, on November 13th, the Chinese Health Commission released its 2019 influenza diagnosis and treatment plan, which differed in several significant ways from their 2018 plan, including in that they changed the recommendation that respiratory patients with negative antigen tests for influenza should not have viral isolation tests. Then, according to reporting by the Wall Street Journal, Cambridge University researchers examining the virus's rate of mutations find that, quote, the virus likely first spread to humans sometime between late September and late November 2019. Finally, in January of 2021, Dr. Shi-Zheng Li publishes one of the first genetic sequences of the virus SARS-CoV-2, and in the same paper, she publishes the gene sequence of RATG13, a virus she had in her freezer that came from a bat in Mozhong, and this is published in a way that obfuscates that it is in fact the same virus she partially sequenced several years prior, BT-CoV-4991, in effect hiding the fact that the virus is connected to a mystery illness suffered by 6 miners back in 2012. Is all of this concrete proof that SARS-CoV-2 escaped from the Wuhan Institute of Virology? No, it isn't. For that, we would need a thorough and unencumbered investigation with full access to the Wuhan Institute of Virology. So why hasn't there been one? Join us next time for part 3 of Origins, Birth of a Pandemic. Origins was written, reported, and edited by me, John Duffy. Our producer was Ray Novoselsky. Field audio recording by Kristen Turo. Original score by Zachary Walter. Origins was an independent project by D.R.A.S.T.I.C and is kept ad-free by listeners like you. Please consider a donation to our Patreon account by visiting patreon.com/double-asterisk media. Follow us on Facebook and on Twitter @*_double.